are massively generated within the gastrointestinal tract. The concentration in the stomach is almost 1,000-fold higher than in other tissues or plasma. And also, we know that reactive species of oxygen contributes to exogenous injury to the gastrointestinal tract mucosa, including the damage induced by anti-inflammatory drugs and also GI infection. This slide summarizes the single transaction pathways which are triggered by infection with the Helicobacter pylori. And even in this cartoon, you can see that reactive oxygen species generated by the bacterial gamma vitamin transferase activate a number of intracellular signaling pathways which stimulate proliferation, invasion, survival, inhibit apoptosis, and these are a lot of events which are relevant to the development of gastric cancer in H. pylori infected subjects. Evidence accumulated over the past years strongly suggests that nutrition, and in particular the antioxidant component of our diet, plays an important role in the prevention and possibly even in the treatment of a number of pathological conditions affecting the gastrointestinal tract. This slide shows that the antioxidant activity introduced in our diet through fruits and vegetables is inversely correlated with the development of gastric cancer. And also, supplementation of our diet with antioxidant micronutrients such as beta-carotene and ascorbic acid facilitates regression of atrophy and intestinal metaplasia, which are two preneoplastic lesions in the stomach, and also inhibit the progression of intestinal metaplasia. And probably dietary antioxidants play a crucial role in the management of GI homeostasis and also in the prevention of gastric neoplasia, or gastrointestinal neoplasia, by scavenging reactive oxygen species and nitrogen-free radicals, by breaking lipid chain peroxidation reaction, inhibiting light oxygen, and reducing the bioavailability of food carcinogens. In the past few years, our lab has been involved in research on the potential beneficial effects of nutraceuticals, and we have studied the effect of a variety of apples, which is typical of our region. And it's interesting to know that apple phenolic compounds represent almost 22% of the total amount of phenolic compounds present in the diet of consumers in the United States. So I will present data generated in our lab showing the effect of an apple polyphenol extract on damage induced to gastric cells by reactive oxygen species and by non-steroidal anti-inflammatory drugs. Here you see that gastric cells treated with xanthine and xanthine oxidase, which generate reactive oxygen species, show a dramatic drop in cell viability. And this drop in cell viability is significantly counteracted by pretreating cells with increasing concentration of an apple polyphenol extract. And the protective effect exerted by this apple polyphenol extract is strictly bound to its antioxidant activity. Here you see that cells treated with the apple polyphenol extract show an almost fourfold increase in the antioxidant intracellular activity. As expected, the antioxidant activity drops following exposure to xanthine and xanthine oxidase to reactive oxygen. Here we see once again that pretreating cells with the protective concentration of apple polyphenol extract, this counteracts the increase in MPH, which is the marker of which is induced by xanthine and xanthine oxidase. And these in vitro experiments were confirmed later on by experiments done in vivo in the rat using as a damaging agent non-steroidal anti-inflammatory drug. Here you see a protective effect of the apple polyphenol extract against indomethacin-induced injury, both at the macroscopic and microscopic level. And this is a more recent data in which we show that the apple polyphenol extract prevents damage induced by acute or chronic administration in the rat. And what is interesting is that the protective effect exerted by the apple polyphenol extract is quantitatively comparable to that obtained with isomeprazole, which is an inhibitor of the protonic pump, so it's an inhibitor of acid secretion. And isomeprazole and other proton pump inhibitors are standard therapeutic options to prevent damage induced by non-steroidal anti-inflammatory drugs in humans. But what is interesting is that the protective effect exerted by the apple polyphenol extract is not accompanied by any alteration in gastric secretion, whereas the protection exerted by isomeprazole is accompanied by a profound inhibition of secretion. And this is clinically relevant because the sustained, long-lasting, and profound inhibition of gastric secretion by these agents is costly and is not without any risk to the patient, with hip fractures, pneumonia, increased susceptibility to gastrointestinal infection. And there is also some paper which suggests an increase in upper GI tract tumors following sustained inhibition of acid formation. Another aspect which has been studied in our lab in collaboration with other laboratories is the role of natural products in the prevention of colorectal cancer. We know that colorectal cancer is the third most common cause of cancer death in Western countries. We know that the Mediterranean diet is associated with the lower incidence of cancer, and we also know that polyphenols are richly represented in food of the Mediterranean diet. And so we first studied whether the apple polyphenol extract exerted any anti-carcinogenic effect in colon cancer cells. And we know that the carcinogenesis is mainly due to an imbalance between cell proliferation on one side and apoptosis or programmed cell death on the other side. And here we see that our apple extract induces apoptosis in two different cell lines. And also another effect which is relevant as to the anti-carcinogenic effect exerted by the apple extract is its demyelinating effect on tumor suppressor genes. We know that in many colon cancers there is a hypermethylation of the promoter region of tumor suppressor gene, which leads to the silencing of the gene and of their effect. And here you see that treating cells with the apple extract leads to the reappearance of the band of unmethylated band of MLH1, which is a tumor suppressor gene. And what is more important is that the demethylating activity exerted by the apple polyphenol extract leads to the re-expression both at the mRNA level and at the protein level of HMLH1. And this is true not only for HMLH1 but also for two other tumor suppressor genes, namely P16 and P14. Here you see the treatment